Hi all, this is Suraj Gopal, and today I'm going to be talking about the Unit 3 lesson on cell structure and organelles in the New York City Department of Ed's Living Environment curriculum. And so this video is specifically geared towards teachers of students with disabilities. And when I looked at this lesson initially, my first reaction was, wow, it's fantastic, but it has a lot of vocabulary. And so I want to dive in and talk to you about some ways to break down the process of vocabulary acquisition and also Regents curriculum for students with disabilities. So I'm going to hit the high notes of the lesson and then go into my supports. So we see that we have this initial question. I skipped over the teacher-facing slides in this. And then we have a lesson objective. We're going to explore structure and function of prokaryotic cells and eukaryotic cells, learn the name, structures, and functions of different organelles with a specific focus on membrane-bound organelles found in eukaryotic cells, and then talk a little bit about the cytoskeleton and the cytosol. So it gets pretty complicated pretty quickly. And as you can see from here, the questions come thick and fast, and they are leveled in a really nice way. But my first impression upon looking at all of these questions in the introduction to modeling is that they were pretty sophisticated. Let me pick out a few examples for you. What are ribosomes and what are the ribosomes responsible for making? In which cells would you find ribosomes? Why is the nucleus considered the big boss organelle? What's the difference between rough and smooth ER? These are questions that would take a few sentences to answer thoroughly, and still the student might be grappling with vocabulary that they've seen for the first time. And so after looking at this and also watching the Amoeba Sisters video, my first resource for you to use is a handy set of guided notes. So what I tried to do in my ground floor section here is take you through what I call mastering the basics of cells. We do need to fill in modern cell theory. We do need to get a handle on the difference between prokaryotic and eukaryotic cells. And specifically, we need to get a handle on the fact that they do share certain characteristics, but the differences are important and crucial to remember. Now, some of this material relates to other units, and as you can see from the Amoeba Sisters lovely video, which I have also pulled up and made a guide to in part, is that they do go back and they talk about homeostasis at the cellular level. And then, of course, this is all a jumping off point to get kind of down and dirty inside cells and understand the organelle. So by answering these handful of questions initially, a student will get a grapple on how to differentiate the two basic types of cells, how to relate them back or their function back to the idea of homeostasis, and then how to take the lens and move in a little bit closer and look at the components of a cell and why they're important. I like to, in my lessons with students with disabilities, I like to always have a section, especially in the remote virtual era of teaching, have a section for student to record questions. What I do frequently on Google Classroom is I have students turn in notes, and these notes are considered participation credit, but they also provide me a valuable entry point for feedback. What I mean by that is if students are recording questions here, in addition to maybe answers that they have that reveal a certain level of mastery or a certain level of struggle with a concept, these questions they write let me know exactly what I'm going to loop back to if I ever pull students to work on things more intensively in a small group or one-on-one. And I don't want them to be stressed, so I always kind of make a point of telling them that. If we go on, I do think it's helpful for all of my students to have a constant visual when we're talking about cells. They're so detailed, the diagrams are a little bit complicated, but I do like them to develop a close relationship to the way that these things are usually rendered in pictures. So we often see, for example, mitochondria is kind of like a bean-shaped organelle with little squiggles inside. We see this kind of loopy sort of highway, if you will, for the endoplasmic reticulum, and I'm going into a little bit of the language of the cell city part of the Amoeba Sisters. We see the nucleus as sort of this big central blob, and so on and so forth. So making those connections between the visual and the function is very important to me. Often those two things are nicely aligned. down here below, and again, this is kind of entry point stuff that I would do for a lot of lessons, but I do think it's important to have a resource on hand for you. I want students to purely define the function of the parts below. Now you'll note that I'm giving them one instruction for all of the organelles that we cover and are important for now. You'll also note that these organelles are ones that are exclusive to, well, not exclusive, they're non-exclusive organelles because they exist both in plant and animal cells. And that's important to me to have those listed before we get into the differences between plant and animal cells. You'll notice also that I have a few additional questions. We want to list the structures inside the nucleus. We want to compare rough and smooth ER because they have tremendous differences in function, and so on. Now, after they name, or I guess define all of the functions of these organelles, they're going to name two main differences between plant and animal cells. I like to keep that in a different section because very often you see Regents questions that are independently focused on the difference between plant and animal cells. So having that information separated, for me, helps me draw attention to it easier, and for a student with disability, it can help them just kind of organize their thoughts. If we go down a little bit further, I've got a handful of resources and tricks. And so this can be edited to your liking. I like this as is, but from time to time, I could see teachers maybe removing some of this vocabulary so it's not overwhelming for a student. And as you can see, some of it might be a little bit more teacher-facing in the way that it's created. So I encourage you to take it and manipulate it as necessary. So the first part of this section is key tips. And these are key tips just that I give for lessons in general. So take your time when you're learning this stuff. You've got a lot of vocabulary, so try to master a little bit at a time. The next thing that I really like to do is I often, when we're starting a vocabulary unit, I have a class already in Quizlet, and I have students be able to come in here and do practice. Sorry, it says ready to share it. Yeah, I can share this through this link as well, so I'm just going to copy that for later. But it is linked in the document already, and as you can see, I have definitions in here. And these definitions I wrote myself, and that's particularly because I've found a lot of definitions from outside sources to be a little bit onerous in terms of the vocabulary that they use. So these definitions are particularly simple and straightforward and designed for students to just practice vocabulary through Quizlet. So they can take little quizzes over and over again until they feel like they've really mastered this vocabulary. Now the final thing that I tip them to do is to keep thinking of a cell as a city. Each organelle has a function, just like the analogs that they make with buildings and workers and power and so on and so forth. And I think we want to keep that at the front of our minds as we learn this new topic. And last but not least, I have support vocabulary that is germane to the Regents questions of the day. And so you always have that to draw on, and students, if they're confused about a question, because there's also this vocabulary lift in the question, they can come back to here and usually find vocabulary that is especially important to the unit, but also shows up in Regents questions very often. And then last but not least, certainly not least, I do think it's really important for students with disabilities to have a set method and strategy for approaching these questions. Mine that I like to promote is problem annotation. So you can go into this document, you can go over the process and model it when approaching one of these questions as a group, and then have students try to take on a question on their own with you watching over their shoulder, sort of like an I do, and then maybe a we do, and then finally a you do with some guidance on our part. Very important for them to get in the habit of underlining the question, boxing vocabulary, and I especially think it's important for them to eliminate information that doesn't seem important. This doesn't always come up, but many Regents questions have extraneous information. So these are my supports for this. I hope you find them to be useful.